Assalamu alaikum. A very good morning to my dear students. So, how are you, students? I hope you all are fine. Okay, stay at home, stay safe. Okay. So let us start our today's topic. I am your biology teacher again from United Public School, Tandu. Okay. In our last class, we have discussed about the activity 2, that is carbon dioxide is necessary for the photosynthesis. Okay. Remember? And in today's class, we will discuss about the light and the photosynthesis. What is light? Light is nothing but sunlight. Okay. And the photosynthesis is, it is a process which is occurring in the plants, as you all know. I hope so. So, in Priestley's time, scientists did not understand about the energy, but later so much was discovered about it. If energy is released when carbon dioxide and water is formed by combining oxygen with carbon and hydrogen, then what about the reverse? Which means that in the Priestley's time they don't know about the energy, but later so many discoveries they have come to know about that the energy is released when the carbon dioxide and water is formed. How they formed? By combining oxygen, carbon dioxide and water. As you can see here, oxygen, by combining oxygen, carbon dioxide and hydrogen, the carbon dioxide and water is formed, and here an energy is released. Okay, this arrow mark indicate that the energy is releasing. So this energy is released. By this energy the plants do, they process the daily activities. Then one question will arrive, what about the rivers? If this is our equation, right? So what happen if the process is reverse? What about forming oxygen again by putting back in the air? तो हम लोग रिवर्स करके कि ऑक्सीजन कैसे फॉर्म कर सकते हैं? by putting it in the air, in the gases. ठीक है. Eventually scientists learned that the energy situation would be also reverse. So finally, after so many problems, the scientists came to learn that the situation would be reverse. It is like reverse by from this, like reverse. ठीक है. This is formation of carbon dioxide and water, right? The in reverse process the oxygen is formed by the reverse process. ठीक है. एंड द ऑक्सीजन फॉर्मेशन वुड यूज अप एनर्जी. फिर ऑक्सीजन फॉर्मेशन क्या होता? वो एनर्जी के फॉर्म में यूज होता. दैट मीन्स इफ प्लांट्स फॉर्म ऑक्सीजन दे हैव टू गेट एनर्जी टू मेक इट पॉसिबल. सो व्हेन द प्लांट फॉर्मिंग ऑक्सीजन दे विल गेट द एनर्जी टू डू देयर डेली प्रोसेस, डेली एक्टिविटीज. ठीक है. वेयर डिड द एनर्जी कम फ्रॉम? वन क्वेश्चन ऑल्सो अराइजिंग हियर, वेयर डिड द एनर्जी विल कम फ्रॉम? लेट अस सी. A Dutch scientist. His name was Jan Ingelhaus. His lifespan is from 1730 to 1799. He found the answer about this question. How it formed? See, he studied the way in which plants form oxygen. He studied the way in which plants is forming oxygen. In 1779, he noticed that in aquatic plant, he noticed that it happened only in the presence of light. In an experiment with the aquatic plant Hydrilla, he observed that in bright sunlight, small bubbles formed around the green parts. So what he observed? He observed that the energy comes from the light, the sunlight. He did an experiment with the aquatic plant. Aquatic plants means water, the plants which is present in water, that is an aquatic plant. And he took a specific plant, that is Hydrilla. He observed that the bright sunlight shows small bubbles formed around the green plant, while in the dark they did not form, which means he took an experiment. He placed the Hydrilla plant and he observed around the Hydrilla plants there is small bubbles. Bright sunlight observes and the small bubbles is forming around the Hydrilla, which means that bubbles containing gases, which means the gases is nothing but oxygen. But while in the plant is placed in dark place, that the bubbles are not forming, they did not form. So by this he also found that the gas present in the bubbles contain gas. The gas is nothing but oxygen. Clear? He just Jan Ingelhaus is just did an experiment. Later it was further confirmed when Engelmann in the early 20th century indigenously detected the point of maximum rate of photosynthesis. Later he confirmed that this experiment was right. So for confirmation test, what did this Engelmann did? He used a stand of algae and ठीक है, and he exposed to directly to the colors of the light. While using sunlight, instead of using sunlight, he directly the algae directly exposed to the lights. The light contain colors, that we have rainbow colors, that you know na rainbow colors, red, blue, grey, red, blue, orange, that colors he exposed. Then he used oxygen sensitive bacteria, okay, and found that they crowd around an area illuminating with red and blue rays of light. So with the help of this oxygen sensitive bacteria, they gave this bacteria gives oxygen to this algae in the light, in the different colors of the light, and they found that the crowd which is creating around the hydrilla plant, aquatic plant, right, those are illuminated with the red and blue light rays. Illuminating means creating light. So these colors of rainbow lights are affecting on this, and it creating a blue red color light rays. These lead to the most studies on effect of light on photosynthesis, right? And the role of different colors compounds are called as pigments in plants, and the utilization of light energy. As you know, the different colors of the compounds present in the plants, right, that is nothing but pigments which is present in the plants. By the pigments of the plants, the color will be shown on in plants, okay, and the utilization of energy is necessary. So we have an experiment, a lab experiment, that is experiment to observe the oxygen is evolved during photosynthesis in the presence of light. As we have discussed this, we have to do an experiment and we have to prove that it is a right, okay. So we have to take some required materials, that is the beaker, funnel, test tube, and hydrilla, and any other aquatic submerged plant. What is meant by submerged plant? Submerged plants means the plant which has roots, rooted plants. The plants is vegetative mass is below the water level. We are using here aquatic plant, right? So vegetative mass means more amount of portion will be below the surface and small portion sticks above the water. So like that plant we have to took and black paper, bucket full of water, an instant stick, mat stick, okay, and we have to do our procedure. By taking these materials, we will start procedure, okay. Arrange the apparatus as shown in figure 5, as you can see over this figure here, and make two identical set of this water plant. We have to take two identical means similar, same set of the water plant, okay. Then take the whole set under a bucket of water and invert the water filled test tube over the stem of the funnel. As you can see here, we have to arrange the like this in this manner, okay. And we have to put the test tube like this. Invert means ulta. ऐसे डालना कि in such a manner that the water should come from up to the stem of the funnel, which means up to some long distance of the funnel, and this helps to retain water, ठीक है. Take the setup out of the bucket and keep it in सनलाइट. पानी में रखने के बाद हम लोग को इसे बाहर निकाल के सनलाइट को एक्सपोज करके रखना. फिर अरेंज द सेकेंड सेट इन द सिमिलर मैनर, कवर इट विद ब्लैक पेपर क्लॉथ, कीप देम अंडर शेड. सो एक सेट का तो हम लोग इसमें पानी में डाल के निकाल के सनलाइट को एक्सपोज कर दिए, ठीक है. फिर नेक्स्ट क्या करना, और एक सेट, दो सेट्स. आई टोल्ड यू दैट वी हैव टू टेक टू आइडेंटिकल सेट्स, राइट. तो दूसरा सेट हम क्या करना है, सेम इसी तरह पानी में रखिए, उसे पानी से बाहर निकाल के क्या करना, उसको कवर कर देना ब्लैक पेपर से. वी हैव टू कवर इट विद ब्लैक पेपर क्लाउड सो दैट इट विल नॉट रिसीव सनलाइट, सो दैट इट विल स्टे इन डार्क प्लेस. Them keep them in shade, which means in shadow place, okay. You would see that in place of water there is no air that will in the set up kept in sun. So which the set one which we expose to sunlight, what will you observe? We will observe that the is filled with a bubbles, the gases, the bubbles. As you can see over here, the test tube is filled with some bubbles. These bubbles have the gases, which means oxygen. The gases is nothing but oxygen. Actually a gas that is collected inside the test tube, ठीक है. ये सन को एक्सपोज करे से उसके अंदर हम लोग को बबल्स ऑब्जर्व करने मिलेगा. बट ऑब्जर्व द अदर सेट, सेकेंड set which we placed in dark side, right. So there is no difference in the amount of gas, so which means they have not created this bubbles. So because of sunlight, it is proving that it is because of sunlight. So we have another idea, that is test the glass in the test tube by inserting a glow matchstick or the intense stick which would burn into the flames. This shows the presence of oxygen. अगर हम लोग कभी भी समझ नहीं आया तो हम लोग क्या करना, ग्लो मैचस्टिक और इंटेंस स्टिक लेना और उसको इसके अंदर डालकर टेस्ट करना. टेस्ट करें तो क्या होता है, अगर टेस्ट ट्यूब इज प्रेजेंट, oxygen is present in test tube, then it immediately catches this matchstick immediately catches the flame, ठीक है. flame, it catches the fire. This shows the presence of oxygen. In presence of oxygen only the matchsticks catches the fire. So if we placed over here the matchstick, then if it catches fire, this means the oxygen is present over this. Okay, by this we have completed. So as you can see over here, this is our beaker, this is our funnel, oxygen bubbles are coming out of here, this is our hydrilla plant water funnel. Okay, all set. So by this we have completed our lab activity, that is hydrilla plant experiment. And what precaution do you need while removing the test tube from the beaker? Discuss with the teacher. So discuss with me. While removing the beaker, test tube from the beaker, we have to careful that these two parts will not get separated. Okay, this I gave answer so that you can understand. Okay. By this experiment, what we have proved? We have proved that the oxygen is evolved during photosynthesis in the presence of light, which means plants created oxygen by the photosynthesis of the light only. If light not present, then plants can't produce oxygen and it can't get energy, it can't do any work. So sunlight plays an important role in photosynthesis of plants in creating oxygen, right? So my dear students, your homework, today's homework was this experiment. You have to draw this diagram, plus you have to draw this diagram, plus you have to do this procedure. No need of material required, just draw the diagram and do this 4 steps of procedure and write another step what you have observed in case of set 1 and in case of second set. Okay, write an answer in your homework and complete your homework by the time. I hope you all have enjoyed my class. Okay, up to that, thank you. Take care students, have a nice day.